Okay, this morning we're talking about urine formation. Yeah. We're going to talk about today about urine formation, and then the next very short video tomorrow will be about hormones, and that's it. So there's lots of stuff kind of to remember, a lot of itty-bitty annoying things. So remember, urine is formed. So what happens is dirty, when we talked about the blood vessels of the kidney. So dirty blood comes in the afferent arterial, and then it goes into this thing called the glomerulus, the glomerular capillaries. And so this is the first place where urine is cleaned, and it's sort of a like, let's clean it all idea. So this is pressure filtration. So blood comes in, it's under pressure, it goes into the glomerulus, and the capillaries in the glomerulus they're kind of porous. They leak more than regular capillaries do. And the pressure, blood pressure, forces all kinds of small molecules, small things that are in the blood. It forces it out in the glomerulus, and it goes into now the nephron, and this structure in the nephron is called the Bowman's capsule. So that thing that's surrounding, it's like a cup that surrounds the glomerulus. The whole thing is called something cup, but I don't remember. It's okay. And so here's the stuff that gets filtered out. Water, because water is small, comes shooting out. Salt ions come shooting out. Glucose, amino acids. Notice these things are all things that your body likes and wants. And then urea and uric acid are pressure filtered out there as well. So actually a lot of good stuff gets filtered out in this first bit of the urine creation process. So we go shooting out, and then the rest of the filtration process, a lot of the rest of the filtration process is now we got to bring the good things back in. And how much of the good stuff do we want back in? So pressure filtration is the first place. Small molecules get filtered out. These are all small molecules. Large molecules like globulins. Remember, globulins are blood proteins. We learned about those in the blood thing. Blood cells and, platelets, all those things, they're too large. All those proteins, all those bits of cells and things, they're too large. So they stay in the blood, in the plasma. So a bunch of water comes out, those small molecules, and then it continues on. The blood continues on here in the efferent arterial, and the pressure pushes the stuff that was filtered out down the nephron. The stuff that was filtered out is called filtrate. So everything inside the nephron and the tubules of the nephron, the lumen of the nephron, is called filtrate. And now if you think about, so there's a lot of water in the filtrate, and a lot of water has come out of the efferent arterial, and it's full of blood proteins and things. It's become a little bit more concentrated. So actually there's a concentration gradient. There starts to be a concentration gradient. And there's kind of two concentration gradients. So there's the concentration gradient between the lumen and this area, that's the extracellular fluid area. So it's the fluid around the blood vessels and around, it's sort of the cells and that fluid around there. And then there's a concentration gradient that goes into the blood. So there's varying concentration gradients between the lumen, the extracellular region, and the blood or the capillaries. So things go in and out based on that. So filtrate continues down into the proximal convoluted tubule. That's this area here. And here what we have is in the walls of the proximal convoluted tubule, there are carrier proteins. And they actively transport, so they use ATP, and they start bringing back the stuff, most of the stuff that we want. So they bring back a whole bunch of glucose, any amino acids, they just pull those back. They don't bring back the urea or the uric acid. So, uric acid is a funny one. Let's just talk about urea, because uric acid actually does kind of come back into the blood, and then later on in the distal convoluted tubule, that's where most of it gets controlled. But the book doesn't really talk about it. I put it in here because it was in my head, but let's not worry about it too much. So let's just worry about urea. So just remember that urea stays in, that's the toxic stuff, it stays in the convoluted tubule, and we bring back glucose, amino acids, and then and sodium. Now, we don't bring back all of the sodium here. This stuff is moving too fast, and we just bring back some of the sodium. We're just starting to recapture the sodium, and we also don't bring back all the water that we want. There's still just a tiny amount of water that comes back. The water, it's not being actively transported here. The water is passively moving here because there is there's a lot of water in the proximal convoluted tubule. A lot of water entered through filtration. So it's very diluted, this proximal convoluted tubule, so there is a concentration gradient into the tissues surrounding the proximal convoluted tubule. So water through osmosis, because there's a concentration gradient or an osmotic gradient going out, will go into the tissue, and then will go into the blood, because the blood there is actually quite concentrated, because a lot of water went out of the blood in the glomerulus into the lumen, into the nephron. So see, there's all these concentration gradients. You have to be kind of mindful, keep track of these concentration gradients, and we talked about all that, and I know it's a little crazy. But just know that, so you got active transport happening here for glucose, amino acids. Water is just passively diffusing or osmosing. So are these chlorines. It doesn't show it on here, but because this sodium Na plus is also attached to a chlorine, but the chlorines here are just moving through passively through channel proteins. So there's going to be channel proteins in the walls of these tubules. So that's where we recapture a bunch of the goodies. So we filtered everything out, then we took back actively the stuff that we wanted. So yeah, words like hypertonic, right? So the blood here is hypertonic to the extracellular fluid. So if it's hypertonic, water wants to go to a hypertonic region. Then we start the major amount of water reabsorption. So because we want to bring that water back, otherwise we just don't want to be we don't want to be drinking water and just peeing it out. We want that water back. We need the water for cellular respiration and for all the things for cooling us and all the things that water does in the body. So we're going to start reabsorbing water. And we do that here in this thing called the loop of Henle. That's this region here. So as the water goes down, the loop of Henle, if you imagine, remember the kidney. So remember, this top region is called, this whole area up near the surface is called the cortex, and then as you go down, it's called the medulla. So the nephron, the Bowman's capsule, and the proximal convoluted tubule are all up here in the cortex, but the loop of Henle goes down into the medulla. And the medulla, the extracellular fluid, the fluid around the loop of Henle, is really salty, way saltier than the fluid in the cortex. So then the extracellular fluid in the cortex. So as the loop of Henle goes down, this area here is super salty. All around here is really salty. I think yeah, there we go. So this area here is really salty. So this is a lot of action going on here in the loop of Henle. So what happens here is there are channel proteins all along the descending arm of the loop of Henle. So things are going down, and it's really salty here. So what's going to happen? It's super hypertonic. So the water is drawn out through osmosis out of the filtrate, and at the bottom of the loop of Henle, the filtrate is at its highest concentration. So it's got the lowest concentration of water, but it's got the highest concentration of solutes. Okay. So it still has salt and stuff in it in the loop of Henle. So it's really, the loop of Henle, so it's really, there's a lot of salt and there'd be potassium and things, but we're not talking about that. But the salt, there'd be a lot of salt in here. So the water gets drawn out because it's really salty here. And there are channels, aquaporins basically, or channel proteins, that passively let H2O go out. Now, as the water is being drawn out, the concentration of salt is being diluted as you go down into the medulla. So here it's actually less salty because there's so much water has been drawn out. It's diluted the extracellular fluid. And here the bottom of the loop of Henle, it's called the nephron loop, is really salty because all the water's been drawn out. So what starts happening is now passively salt. Those salts can be drawn out. They can diffuse out because it's less salty here because all this water was drawn out by all the salt. See, it kind of works backwards. It's kind of crazy. So here the salt starts being drawn out, and now the concentration of salt in the filtrate is less, and there are no more aquaporins, or there are no more channels for water on the ascending arm of the loop of Henle. So water can't get out. But now what's happening is there are pumps as you go higher. There are pumps now. So the first bit, the salt is just passively being drawn out through diffusion. And so it gets drawn out passively, so the concentration of salt goes down inside the filtrate, and then active transport picks it up, and it starts drawing the salt out of the filtrate. And this salt that it's drawing out enters the extracellular fluid, and it makes the extracellular fluid really salty, which draws out the water on the descending bit. So you see it's sort of fueling itself. First it draws out the water, then it draws out the salt, and then it pumps the salt to draw out the water, which will draw out some salt, which it pumps the salt to draw out the water. And it's sort of this, I don't want to say it's not really a cycle, but it's kind of this neat pump. So filtrate goes on. Now it's had the salt removed, not all the salt, it's still got some salt in it, and it moves up back into the cortex. It's not as salty up here. The extracellular fluid is not salty here, and there's not that much salt in the distal convoluted tubule. And so what happens here in the distal convoluted tubule is you have excretion and secretion. So if you have any like drugs in your system, penicillin, whatever, I guess THC would be something like that. Maybe, I don't know. Actually, don't quote me on that. So penicillin, any histamines, things like vitamins, excess vitamins. You are a total health store nut. You buy packs of vitamin B and vitamin C, and now it's full in your blood. Well, your body's not going to use all that stuff. What it's going to do is it's going to put it into your pee here. So there are proteins, channels and active proteins on the outside there, and they will pump, they will excrete or secrete, sorry, these substances out into the filtrate to go out in your pee. The other thing that happens at the distal convoluted tubule is the pH of blood is controlled. So if your blood is too acidic, what will happen is those extra hydrogen ions will be excreted, into the filtrate, out of the blood. So there's blood vessels that run along there, and they will, there we go, there's drugs, and hydrogen ions get excreted, and bicarbonate ions get pulled out. So bicarbonate ions actually did end up in this filtrate too, and they will be pulled into the blood to make the blood more basic, and the hydrogens will become excreted. And the reverse will happen. Bicarbonate ions, if your blood is too basic, bicarbonate ions will go into the filtrate, and hydrogen ions will get pulled out. So pH control and drugs, excess drugs and vitamins go here in the distal convoluted tubule. and finally into the collecting duct. So this is the collecting duct, and the collecting duct leads right to what's this renal pelvis area. So this is the final place of pee, and what happens is the collecting duct is going down again into the medulla. Remember, the medulla is all salty because we pulled out all of that sodium, sodium chlorine. We pulled out all that salt. So the walls of this duct are actually porous, and they're actually controlled by a hormone, which we'll talk about tomorrow, and water osmosis out of the collecting duct. This is where a lot of water gets pulled out. As well, a little bit of urea gets pulled out, but this urea gets pulled out and then it just goes back in to the convolution, and it's just a tiny bit leaks out. But the rest of it, most of the urea goes out into the renal pelvis, down the ureter, into your bladder, out your urethra, and you pee. Okay, that is urine formation. So just remember what happens at each of those, what kind of urine formation happens. So at the glomerulus, at the Bowman's capsule, you have pressure filtration. You have those small molecules come out. Then you have the proximal convoluted tubule. It's all in the cortex, this bit, and you've got active transport to put glucose, amino acids, to pull those things back, the things that you want. Water also starts diffusing back out there. Then you start going down into the medulla in the loop of Henle. First, all the water is drawn out into the salty medulla. Then salt starts to get drawn out and then pumped out to make the medulla very salty. Then you go up to the distal convoluted tubule. You've got transport, and you've got secreting and excreting. You've got pulling drugs into the filtrate, into the urine, and you've got pH control. So you're pulling out hydrogens, or you're dumping out hydrogens. You're pulling out bicarbonate, or you're dumping in bicarbonate. Then finally, in the collecting duct, you are collecting the rest of the water out of the urine, or as much as you can. There's always going to be a little bit of water in the urine. There's water in the urine. But you're drawing back the remainder of water in the collecting duct. And that is urine formation. Okay, talk to you soon.